Değerli katılımcılar, hepinize hoş geldin diyoruz. Bugün İstanbul Ticaret Üniversitesi'nin 11.sini düzenlediğimiz Science and Society, Toplum ve Bilim buluşmalarına iştirak ediyorsunuz. Bugün hakikaten alanında duayen, dünya çapında isim yapmış bir hocayı ağırlıyoruz. İlhan Fuat Akyıldız hocamızı. Büyük şeref duyuyoruz. Kendisi kabul etti. Üstelik saat noktasında da bir hata yaptık. Biz onu da itiraf etmemiz lazım. Şu anda Amerika'da saat 7. Hocam sağ olsun bizi kırmadı. Bilgisayarının başına geçti. Ve bizi aydınlatacak. Çok önemli de bir konuyu irdeleyecek. Biliyorsunuz bu Internet of Things kavramı 10 yıllardır aslında bir research alanı. Ama hocam bunu bir adım öteye götürmüş vaziyette. Nanoteknolojiyle birleştirerek biological nano-things kavramı üzerinde bugün bize bir konuşma yapacak ve bu noktada hastalıklarla ilgili nasıl müdahale edebiliyor bu cihazları kullanarak bunu anlatacak. Ben sözü çok uzatmayacağım. Hocam varken bize laf düşmez hakikaten. Öncelikle kendisine hakikaten katıldıkları için çok teşekkür ederiz. Bizi kırmadılar. Ve ümit ederiz ki bu pandemi süreci bittikten sonra da Türkiye'de üniversitemizde ağırlamayı, Boğaz'da birlikte güzel bir yemek yemeyi ümit ederiz. Ben sözü kendisine bırakıyorum. Tekrar şeref duyduk katıldıkları için. Saygılar sunarım. Buyurun.
Günaydın veya iyi günler herkese. Öncelikle Zaim Hoca'ya çok teşekkür ederim beni düşünüp davet ettiği için. Maalesef bu pandemi dolayısıyla fiziksel orada olamadım. İnşallah Zaim Hoca'nın dediği gibi yakın zamanda oraya gelip hem sizle tanışır hem de dediği gibi Boğaz'da güzel yemek yeriz. Zaten İstanbul gibi dünyada hakikaten başka şehir yok. Ülkenin ve şehrinizin değerini bilin yani. Maalesef ben İngilizce sunumu vereceğim çünkü Türkçe çok zor oluyor bana bu deyimler ve akış için. Onun için özür dileyim. The topic or the title of the talk is Internet of Bionan Othings, especially how to detect and avoid infectious diseases. So I started this research back in 2006 about nanoscale machines. And I coined the terminology Internet of Nanothings back in 2008. And then we realized that we can also use the molecular communication with special bionanosensors. And then we also coined the terminology Internet of Bionanothings. We published them. And so we did a lot of research. I created centers in different parts of the world, like Barcelona, Politecnico de Catalunya, and then in Finland, Tampere. I received Finnish distinguished professor funding from the Finnish Academy of Science. And then I did center in my alma mater from Humboldt Award. So, you know, many people worked on this stuff. Also in America, I received many projects from Army and from NSF. And you can see here some references, particular for this case. So as you see, the majority of this work is just published in July 2020 in the IEEE Access Journal and Open Access Journal. You see all these people, they were professors and postdocs. You recognize some of them are, many of them are Turkish actually. Ülkü is now a postdoc. No, excuse me, she was postdoc here, but she's assistant professor at Worcester Polytechnic in Boston. Tevide is the postdoc of Fatih here. Fatih is a professor, associate professor at Georgia Tech. Bige was my former PhD student. She just finished. She will be assistant professor at Michigan State University. And Phil is the medical doctor from the Emory University. My son was a professor here. He left. He has his own company here. He's from Iran. So Phil is American, as you see. So it's in the United Nations, typical America, right? So now here, this paper was published back in 2015, although, as I told you, all this concept started back in 2006-07. And this was Massimiliano was my PhD student. He graduated in 2013. He's at University of Nebraska Lincoln, and he's from Italy. I hired him from Milano of 2008. And Sasi and Yevgeny were people or professors in the Tampere when I received this Finnish distinguished professor position and large grant. So I had that team there. And then here you can see there's a booklet that we published with Massimiliano. So, this third one has all the details. And then, the introduction is from my former papers and from this one. And then the technical part will be from the first paper. So you can see this picture that it will explain you about what is the Internet of Bionan Othings concept. So you see this human body has all these kind of like biological paradigms, like neurons, hormones, and calcium ions, calcium signaling, molecular motors, bacteria, and there are zillions of them, and they create these cooperation, and then they communicate with each other inside the body. And then what we are trying to do is we will try to mimic all of these. We will create artificial, nanoscale machines. Actually, these are very small scale paradigms here. And so we will try to create these artificial nanoscale machines and inject them like artificial bacteria, for example, or artificial molecular motors, or program. You can also call them program. And then they will be helping us to attack many, many diseases. So I will explain that more to you. So now what happens is while these inside of the body has all these biological phenomena, and they communicate, and then we can also try to get the information out of the body. Like we will have these gateways, interfaces. We also call them bio-cyber interface. They're like tattoo type that you can put somewhere on your shoulder or on your arms. You can have one or multiple, and it can use near field communication to your cell phone because it's close to you, right, most of the time. And then you can use the backbone internet, and then it can reach the physicians or healthcare providers, doctors, in other words. And then doctors can remotely reprogram them, right? So you can transmit the queries or commands, reprogram these. You will see them, and then by this way, it will be two-way communication. Of course, mostly the communication goes from the humans to the physicians, right? More asymmetrical. And as you see here, that's the Internet of Bionan Othings concept, right? So we have the backbone internet and then the human body inside. Here's another thing, that we have two different worlds. One is the biological, chemical, or molecular world, we call it. They're using molecular communication. And this part uses the electromagnetic communication. So there is also a mismatch, right? So these gateways will take care of that, so that we can go from molecular world to the EM, electromagnetic world, or vice versa, right? So let's go through this biology basics. Maybe some of you took that in your high school, or also in the college. So what are these Bionan things in the human body, right? So there are human cells are called eukaryotic cells, as you see here. And then the bacteria type here, as you see, typical E. coli bacteria, they are called prokaryotic cells. They are typical nanoscale machines, natural nanoscale machines inside the body. And then, as you see here on the left-hand side, this is a eukaryotic cell tissue. You can see that all these cells, or eukaryotic cells, billions of them, they communicate, interact with each other. They can also cooperate, and they can create a colony, right? And here the same way, the bacteria populates, and then they create this, it's called quorum sensing. So they multiply themselves, and they can also interact and then also cooperate. That's really happening in the inside the, in the nature, in other words. So when we look at the inside of these biological nanomachines, you can see that they're perfect little computers or nanoscale machines. Like here, this nucleus can be used as a processor, and the ribosomes are like biological memory. And then you can see here, mitochondria is the energy source for each cell. You can have multiple mitochondria here, or you can have chemical receptors like these. They're like acting like biological sensors or molecular receivers. And then you have these gap junctions, and they can be regarded as molecular transmitters, and so that's the typical eukaryotic cell case. And this is a well-designed eukaryotic cells. The prokaryotic cells, like the E. coli bacteria, is more irregular behavior, structure, as you see here, but the same thing, like little computer, right? So it has this nucleus and plasmids. Okay, it's like can be seen as a memory. Like you can see here, memory. You can store communication functions, sensing functions, and actuation functions. So you can program them, as I told you before. So we can have coding, and then we can code them into this memory, and then they can do what we want. And so then also these plasmids, they can be regarded as processors. These are the pilies, and they act like sensing and actuating. And this flagellum, like a tail, is like an actuator. It navigates the bacteria, and they're extremely slow moving. Like 1 centimeter, they need like 4.5 hours for 1 centimeter. Plus, they're always tumbling, so slow. And what you do is you feed them with sugar, they grow exponentially. And then it's moist area, of course. They move around, and then if you don't feed them, they don't have enough sugar or glucose, then they die. So that's the typical bacteria behavior. Then there are these receptors and membrane, cell membrane. They are like nano-power unit, right? Energy sources. So now this is a typical mitochondria under the microscope. This is a real picture here. So I told you the energy source for the cells, for prokaryotic or eukaryotic cells, especially eukaryotic cells, is the mitochondria. So what happens is that they combine the glucose, amino acid, fatty acids, and oxygens, and synthesize as ATPs or adenosine triphosphate. So now by this way, the energy is loaded, harvested, you can say that. For example, so when you are tired, you say, oh, let me go and get fresh air. So that's typical, you need oxygen, right? So that you can, charge your mitochondria. Or when you have little kids, the doctors tell you do not give chocolate or sugar before they go to bed, because all the mitochondria will be charged, they will be hyperactive, right? So that's the typical example for the energy sources. Then you can see also for the biological memory and processor, like you can see these DNA strains, right? And DNA strain in the nucleus contains the information of protein structure. Like it can be seen like this. Some of you may know this Turing machine, right? One-band Turing machine. So like that's the memory, right? And the ribosomes read and process the DNA information from here and synthesize the proteins, okay? And the proteins, like looks like this, control the functionalities of a cell. Like for example, For transmitting, it's called cell signaling, or receiving, it's called ligand binding or chemical binding, right? So these are the terminologies that the biologists and chemistry people use. So as I always kept telling you that there is communication between these cells. Like here is an example, eukaryotic cells, they release these molecules, as you see here, like they can be proteins, ions, and hormones, and like they flow, and then, you know, normally they're in confined area, right, inside the body, but you can also see that that's like, if you want to see as a model, you can also do free diffusion, right? Free space. But anyhow, but this is the, I'm trying to explain the basic ideas about how molecules are flowing, and the molecules can carry messages, right? And then the receiver will receive them. You will see the process. And then the prokaryotic cells are like this. So same thing, like this is a transmitter and receiver, and it releases these molecules, for example, auto-inducer exchange for quorum sensing. Or there is also another way of doing communication is that, so like this bacteria will upload the information from this device, any other nanoscale device, or any type of device as transmitter, and it will be loaded up to this plasmid, and then it moves slowly, and then it come to the to this one, and then or to this one, and then it can upload the information from here to this one, kind of like handing over the information to the next one, and then finally it reaches the destination, right? And this process that you know, one bacteria gives to another bacteria the contents of its plasmid is called chemotaxis process. And then when they reach the destination, as you see here, they eliminate. Indeed, under the microscope, you can see that. So it's called also mating, that the bacteria are mating. They are very happy that there is a elimination, you know, right, like the light. And so that process is called conjugation. So this is the typical, that's what's happening in the nature, by the way. So now you can see also in this picture how the transmitter looks like. Like here is this one eukaryotic cell, and all these molecules, or you know, if you have one type of molecule, you can call them particles. They diffuse, okay? And that's why you can use Brownian motion to model them, or diffusion solutions from Adolf Fick's physics laws 1 and 2 to understand and mathematically model these processes. That's what we did, and other people do that too. So now they're releasing, and they have some messages on them, right? And then they flow. And of course, some of them you lose, that they are typical losses. Some of them will reach the receiver. And then as you see here, when they reach the receiver and they are sucked by the receiver here, as you see here, by the gap junction, and that's called particle binding or ligand receptor binding process. So, you know, like their binding, you know, are received by the receiving end. So this is the typical behavior of the receiver and in the molecular communication. And that's why these are called also chemical receptors, right? And then some of them, of course, they cannot reach, they move somewhere else, right? So, or just they're lost. So these are the typical transmitter and receiver modeling of these eukaryotic and prokaryotic cells. So now the question here is, or was in the past, can we create man-made biological nanomachines, right? So I explained so far the nature. So can we create these man-made, you know, programmable or genetically engineer these machines? The answer is yes. And we can do DNA manipulations and genetically engineer them. So that's the good news, right? So now you can see here the prokaryotic cells, the bacteria is well understood. So we can genetically engineer bacteria without any problem. We can also have, we have codes, we can program them and they can do whatever we want. So that's really like well understood, and people are applying this. For example, I have many applications here. One of them I remember, I don't have the slides. There is so-called Crohn's disease, an awful disease for the intestines. So there's no cure for it. And there's a bacteria-based disease. You have to go to toilet all the time. You can never cure, unfortunately. But with this technology, people are trying to use genetically engineered bacteria, like healthy ones, and they inject inside the body so that the healthy ones will fight the sick ones or ill ones. And the probability is very high. For the timing, what they do is they take the feces of a healthy person, and then they have these healthy bacteria from a healthy person, and they convert them into pills, and then people are taking them. But that's really only 20-30% success rate. But with this, we are hoping that you will get very high cure rates. Not, this is just an example. Any bacteria-based type diseases, we can fix them, right? So now the human cell engineering is really not easy. Otherwise, it will be fantastic, right? So we can, solve all the illnesses in the human body. So now, how they can try to do is they try to insert a piece of new DNA code into so-called lentivirus cell. This is a special type of virus using genetic engineering. And this lentivirus infects the eukaryotic cell, the healthy cell, and injects a new DNA code into the cell DNA right here. And then it replicates itself, and then it will bind itself here, and then it can go and attack these, whatever the problems are. So, and this is still ongoing issue by these bioengineering medical people, etc. Right? So now you can see the objective here is we try to create man-made machines, like these or these here, right? Like, man-made meaning, genetically engineered. And so the origins go back 100 years ago by Paul Ehrlich. He was an Austrian doctor, medical doctor. He created this concept of magic bullets, in German magische Kugeln. And so he received Nobel Prize in 1908. These agents or these bullets will go and kill the targeted diseases, and without affecting the other healthy parts of the body, right? Otherwise, you will die if it has all the negative side effects, right? So what is the lessons we will learn and get? So this picture was done in 2012 in Finland. There is a newspaper called Aalmuhti or whatever, Good Morning or something. And they had 6 pages report about that center that I created that time. And so the guy made a photo of mine, and then he put me at the wall, and then he put this in the newspaper. So he had all this in Finnish, of course, right? All these things. So now the objective here is the following. We are hoping that we can have solutions for Alzheimer's, epilepsy, depressions, then brain cell monitorings, any tumors you can have, and diabetes, for example, heart diseases. We can always monitor them with special, special design and program these devices. And then, cancer monitoring devices, all of that. Again, depending again what you are trying to monitor, right? And so you can inject these magische Kugeln, or now they're called nanoscale Bionan Othings, and then they can observe what's going on. So as you see, let's say you have heart disease problem, They can always monitor it. And if there is any alarm, they can try to solve themselves. If not, then they can alarm to outside through using this gateway and then going to the physician, and physician can reprogram them, etc. Right? So this is really a breakthrough that that can extend the lifespan of humans a couple hundred years, right? But I assume we'll have some other problems, like here, this COVID-19, right? It was expected, by the way. But anyhow, let's continue. So now we also did a lot of work with my former student Youssef Shahibi. He was from Morocco. Now he works for a company, and Massimiliano is professor, as I told you. And this guy was from Samsung, a Korean guy. We try to create this model for particular drug delivery system. So you can see that normally, I'm sure you learned this in the systems control theory or whatever. So you have these boxes, right? Input, processing inside the box, and then outside, right? Delivery. So in other words, you inject these drugs, medicine, and they travel through the body, right? And then, like here, you can create, and then they go to the target area, and then, they try to attack those, ill areas to have cures for them, right? So the issue is the following. So now here's the target that, once these drugs reach this target, they lose their strengths because they travel through the body, right? And then as they travel, they lose their strengths. And then once they reach here, they're almost like half strengths, having half strengths to reach this target location, right? So now in this study, and also as you see here, this is kind of a communication network, right? Like a propagation, all these particles, drug particles are moving inside the body, right? It's kind of like a communication network. So we developed these as a communication network. And then this was a transmitter that we are trying to inject these drugs. And this is the receiver, meaning the target location will get these particles. So now that was the model, but then what we did is we tried to do it the other way around. That means we want to have 100% strength here, right? And then we go backwards and try to find, usually in the systems theory, what you do is control theory, input inside the box, you try to optimize the output, right? So we did exactly the opposite. So the output is the optimum value for this strength of the drug. And then we try to find out backwards how much drug particles we should inject inside into the body so that we will get the highest degree of the strength for the drug here. So that was really a good paper that we published some years ago. And then we also received this, I call this Monaco project, molecular nanoscale communication. These were professors in Georgia Tech. He's in info theory, and this guy was from mechanical engineering. This guy was from biology. This was from computer science. So I created this team, and we got 5 million in 5 years, 11 to 16. We had like, 20, 25 people working on the project. And although it's over now, so the project page is still there, we published many, many papers, and those papers got a lot of citations. So they were really interesting. And still people are using those, bacterial communication. What we did is, so the idea was, we tried to establish theoretical foundations of molecular communication networks, understanding how they communicate, and try to map them into the communication networks, communication theory. And we developed this fundamental base for that. and for the bacteria-based communication. And then we looked at network architectures like modulation schemes and communication protocols. And also we were published them, modulation, medium access control, etc. And also like the collisions, right? All these bacteria collide, by the way, and losses and all that. And then, so the NSF wanted that we will implement everything NS3, which is a publicly available simulation tool. So we also did that part. So this was all theory, like academic. And then the other team, the mechanical engineering and also the biology guy, we did physical experiments with actual bacteria. Of course, here in the top theory, you can do anything, right? You can conquer the world. But when you have these physical experiments, then you have to reduce all the theory so that you can have some experiments to validate some of these theoretical solutions. And that was quite successful. As I told you, we have many publications. So now, and then after 2017, I wanted to do some more, and then I created a new team. It's always better to have a new team, right? So I created this team that I talked to you about it, and then we wanted to have an application, right? And then we called this Panacea. Panacea means in English, a remedy for everything. That's a Latin word. I found it at 3 o'clock in the morning when I was sleeping. We should call it Panacea. And so then, we started in 2017 with all these meetings and projects, papers, and then finally we published that. So it takes forever, when you have all these members. So now the issue is, how can we detect infections early for critical patients, like patients in critical conditions are better. Like, for example, the cancer patient, like chemo patients, they're very vulnerable to serious bacterial infections, or cystic fibrosis for the young children. Infections come wave by wave for these young children. And then, so our solution should somehow early detect them, mitigate them, right? Eliminate them. So in other words, we try to continuously monitor of these bacterial infections and take actions, right? Early detection, timely administration of antibiotics, and also tracking efficiency of the antibiotics. That means overall, we try to reduce the mortality and also the hospital costs, right? So like I remember, it's a bacteria base, by the way, diphtheria, like kuş palazı in Turkish. And I had it, I was 4 years old. And those years, Turkey was not like now, right? So I remember there was another little fellow next to me, Musa was his name. He died. I remember the parents were crying, Musa, don't die, Musa, don't die, in Turkish, of course. And so then doctors told my parents that, either Allah will help or he will die too. And then somehow out of blue, this thing came out, this black thing, from my throat. Kuş palazı is like, it's choking you, right? It's typical bacteria base, So really, it's a serious thing, as you know. And now you have shots and aşı, etc. But still. So now what we are trying to do is, so this is the internet of Bionan Othings, right? So this part is the human body, like physical world. And you can see here, this word also remor, I came up with that in the 3 o'clock in the morning, like istihare yatıp having some ideas, right? No, but anyhow, remor means explorer. So these are Bionan things, like Bionan sensors, which we will inject inside the body so that they can monitor the, all these, of course they will program, and they can monitor these bacteria's diseases, right? And then you see, remember, we had these gateways, and near field communication through cell phone, we go to the cyberspace, right? Cyberspace, this part, right? Cyber physical space, right? So physical space is here, cyberspace, all this data will be collected, right? They can be aggregated when they have similar data. we'll process them. And then we have to, of course, consider the security aspects, the privacy issues. And then we will use, I will explain you more, some machine learning algorithms to handle all these collected data. And then you can see, like, CDC is the Central Disease Control here in Atlanta for the entire USA. And, you know, we can help them here, or clinic databases or CDC databases. So that's the closed loop of the Internet of Bionan Othings concept, right? And so this is the entire project that we had. You can see we have the Trust One physical domain, like this part, right? So we have these bio-nanosensors, like hardware design. And then, of course, we have all the interfaces. You will see them, and integration of these different components of these bio-nanosensors. And then also look at the physical layer hardware security, right? And then the communication among these hardware devices that we developed, like how do we model, that's more communication theory, right? Infections and drug deliveries, and then how do we realize the near field wireless and power transfer between the Bionan Othings to the gateway, and then from the gateway to the cell phone, and cell phone to the backbone network here, right? All the communication aspects. And then there's the cyber domain with all these, you know, computer science type research. How do you collect, aggregate data? And then how do you use machine learning or deep learning or any type of learning algorithms to help us to early detect these infections and process them and then take actions, right? And then actions meaning, for example, deliver some antibiotics or drug delivery, or also visualization of the problems. Of course, all the security and privacy are important when we do all of that. And then, you know, these are all the trusts or work packages, as Europeans say. And then here are the experimental validations, testing individual components, then prototyping Panacea in an in vitro and also in vivo. So that's why we had these, you know, not only 5 professors, and then we had also like 10 different people working on this. So what are the challenges when we look at the cyberspace, right here, this part, right? That's typical all computer science related research. So how do we effectively collect data and deliver to the, you know, computers or cloud, or, you know, to the users? And we may have energy and transmission constraints. We have to consider those. And bacteria concentration reflecting infections may vary from patient to patient. So that means, how can we early detect these possible infections with high accuracy? And then also, how can we quickly detect those infections and with high accuracy, right? That's also important, because if it's too late, then it's too late. So then patient health data is very sensitive, right? That means, how can we relay or transmit these data securely and privately to the physicians, for example, or the clinics, right? Oops. And so then here is more. How do we collect, aggregate sensing data here? Deep learning for early detection, learning-driven drug delivery. We can have active drug delivery, meaning automated antibiotic and enzyme delivery by implanted or dressing devices, or passive drug delivery, meaning alerted patients or patient takes antibiotics orally by the suggestion of the Panacea automatically, or by the physicians remotely using this Internet of Bionan Othings platform. And how do we visualize a reliable platform to access and analyze data? As you see here, it's all data processing on this side, right? And then we also must look at the hardware embedded security issues, authentication, confidentiality, integrity, etc. And then also develop privacy preserving data storage and processing capabilities, right? So now you can see here that physical domain. I'm going down now about these remor. So remor has, we could also call this Bionan thing, right? So it has these two parts. That's called Bionan sensor. Like they have, like they can sense quorum molecules, and there are these reporter bacteria, membranes here, and antigens and antibody here. I will explain them to you more about the details of this. So that's the infected area, for example, because infected area, these bacteria are releasing these molecules, right? And these sensor must somehow capture those, process them, and send this to the interface here. And that's, again, I will also explain that to you. So these together makes one Bionan thing, like remor. And this is the skin. We're using magnetic induction communication to the gateway that we had this, like either the like tattoo type device, like here, Band-Aid, or a watch, right? And it goes to the near field communication to the cell phone, and then cell phone can use the internet backbone, transmit to these doctors, etc., clinics, right? So now let's look at the like more detail here in this part, right? So that's more technical. So what we have here is, in this case, we have, so we have three different sensing types that we are, or sensing units of the Bionan things that we are planning to design and implement. So one of them is called electromechanical sensor, as you see here. So there are functionalized electrodes directly bind with quorum sensing molecules, like here, right? So that's the first one. The second one, it's a bacterial sensor, right, or sensing unit. An engineered reporter, like here, some of these, I showed you before, like bacteria, reporter bacteria like this, okay? And produces an electroactive product upon reception of these quorum sensing molecules, okay? And this electromechanical sensor generates currents corresponding to the concentration of the quorum sensing received. And then, you know, that there, again, you can see binding process, ligand binding. So they're binding or received by this transducer here. And then the third one is called bacterial sensor, okay? So an engineer reporter, bacteria produces bioluminescence, right, like these photodiodes, or fluorescence upon reception of this quorum sensing molecules are coming here, and they're exciting. This is a porous membrane. They have these holes, and these bacteria can pass through, and the bacterial sensor will capture them, right? And then we apply these, as I told you, bioluminescence or fluorescence, right? And then a light sensor, like here, generates currents, right, which correspond to the concentration of the quorum sensing received, okay? So now what happens is we maintain live bacteria inside the sensor here is difficult, and the sensing process is noisy. So that's one of our challenges, that we are trying to solve this problem. So, you know, that's the, you know, keeping the bacteria alive in this, inside the sensor is the biggest challenge here. So now the second case is the designing of the sensor interface electronics, right? Readout chip, like here, sorry. As you see, I mentioned this here, readout chips here. So now we can have these electronics to process signals. I told you that we are using the magnetic induction, like these are the coil antennas. There's the skin, and then on the other side, on top of the skin, that the Band-Aid type or tattoo type thing has all these components inside, right? Like typical, like passive device that can receive this from the readout chip. And then we can also have these wireless simultaneous power and data transfers. And then what we have here is in this first one is 3D rendering of a solenoid coil and showing its key geometrical parameters, meaning how many turns do we have? What is the diameter of these turns? Like all of this needs to be optimized, right? Determined. And then we have this B lumped RLC, the resistor, inductor, and capacitors, right? Model of a solenoid coil and showing its parasitic resistance and capacitance. And the third one is the example bonding wire coil for the variable hub. So we have also this, these are all physical part, right? And then I told you this before. So this part I explain, and then they go out, and then they go through the backbone to the doctors and physicians and clinics, right? So now the IOBN components, we can again model them for the communication aspects. We are in the middle now for these trust two. So we have this like typical transmitter, transmission process, reception process, and the propagation, right? The channel or the communication network, right? So these bacterial infection, these molecules are coming out, and and then they move right inside the body, and then inside, right? And then the remor, which is close to it, I showed you before, right here. This remor unit, I'm showing you this part, right? So now, like from here, transmitter moves here, and then it will be received by these remors, right? And then the reception process. So, and so then we developed these molecular communication channel model and analyzed it. And then we realized that this typical communication theory, right? The channels are time varying, and there is this diffusion process, right? All these bacteria is flowing around within cells or between the cells, and there are shadowing by the tissue structures, shadowing problem. And then there are also multi-path effects, like typical wireless channel effects, right? And you can see here, we can also use them as a MIMO type communication, like MIMO multiple input, multiple output. Like this is one side infected, another side, it's kind of like two areas, like two antennas. And then these are the two receiving ends, like remors, like 2 by 2, right? So, and then we can, we analyze this also, understanding this. So experimental validation, we had testing of these remor components, each individual bionano sensors and also the chips. We tested the variable hubs. We also tested communication protocols. And then we tested all these aggregation algorithms on the cyber area, the learning engines, like deep learning engines, and closed loop with passive delivery, like transmitting these drugs into the body, right? Passive drug delivery, and also security protocols, etc. Right? So the simple, experiments we did here, as you see here, that we had control release of quorum sensing molecules into these body type fluid here. We place this remor inside this tube here, so that this remor will measure these molecules which were infected or not infected. That's what we are trying to find out. And that remor, the bionano sensor, send the information to the, let's say, laptop, and display the the value, or is the bacteria is infected or not, for example. And then we also try to mimic the human body. You see a steak here. That doesn't mean that's a real. So we try to mimic the human flesh, right? Flesh, so that we had this like skin, right? The human body. So now same concept. So now it goes through the human body, right? Through this meat, and then through a variable hub, and then reaches this here. Again, try to measure the quorum sensing molecule concentrations and find out from the concentration level about the bacteria status, right? And then we also did experimental validation in vivo tespit. My son Konulu professor, he had these with Ülkü, these rats. So we had no infection. We put remorse. Tried to, a rat without any infections, and then we put some sublethal dose, doesn't kill but it's infected. We put also the remor here, and then lethal dose to another animal, and then so you can call it animal cruelty, right? So then you can we try to get the information from these remors through this to the backbone network, and then we analyze this. And it worked, it's validated. So now, 2000, last November, I tried to get, because I work with Norway, Norwegian University of Science and Technology in Trondheim, and University of Oslo Hospital since at least 8-9 years now. So I said, why don't I have another smaller version of this project there? We call it Clipeos. And we submitted this proposal back in November. And then in March, we received that we will receive $1.8 million for 4 years. And so now, of course, everybody talks about now diseases, right? All these viruses, etc. Now, a lot of people are start to work on these issues. So I had this since many years, right? These ideas. So now, although this is mostly bacteria-based, like remor will be called gladio, because these will have different design. We actually we need people. So if some of you are interested in Turkey, we can hire you to Norway. So we need new design for the gladio. That's like remor, but new design for hardware, and also the new way of capturing these diseases. But we also want to move to the, of course, to the virus cases, right? So virus cases are, I think I should be faster because I have a lot of things to tell you. So now, first application is like all these things I explained to you was more generic, like how do we detect diseases? Like pneumonia is one of the awful diseases, right, in immunocompromised patients. So how can we detect that? So they have this Pseudomonas aeruginosa bacteria. That causes the pneumonia. So they have these infections, right? Hospital-acquired infections, cystic fibrosis or burn patients, chemo patients, right? And unfortunately, they are very resistant to a large spectrum of antibiotics. And also it can infect other organs such as lungs, urinary tract, kidneys, and skins, right? So now, same concept that we are trying to do for this, pneumonia detection. And so this was explained in Bige's thesis in detail. She graduated in June 2020. I told you she's going to Michigan State University. There's a huge group, electrical engineering and bioengineering, so she will be working there. And so she tried to detect these Pseudomonas aeruginosa bacteria disease like pneumonia. And these bacteria have unique quorum sensing molecules, right? They have these C12 homoserine lactones and N-butyryl homoserine lactones, like these cooperation or quorum sensing molecules. And these molecules are found in bodily fluids such as sputum and blood in detectable concentration. So now, how can we design these remors or program them such a way we can detect these modules, right? That's the question. And then what we did is we tried to, again, model this processing, right, as a communication network, like the bacterial density, transmission process, propagation inside the body, and then reception process, right? So we modeled this, and then we have this propagation process is a typical diffusion process in porous medium. So now you can see here, this figure shows the diffusion process in porous medium. So the black dots are representing the cells, right? And they have these gaps or parts that the molecules will flow through these pores. Like, if we will not have these black dots, it will be straightforward, right? It will be much faster, as you can see here. But unfortunately, it's not the case inside the body. So they will be moving like this, right? And it's also, this area is wet inside the body, right? Wet medium, it's called. So then the molecules are flowing through these pores. And then we are showing this direct flow versus the reality. And it takes much longer, as you can see here. So we have this equation where C stands for concentration of the quorum sensing molecule in the tissue. T is the time, diffusion coefficients, tortuosity. These are all different for the each of the organs. Tortuosity and also the volume fractions, alphas. And F of C is, again, alphas and lambdas are all different for each of these organs and also each person, right? And then the V is the velocity or nu, and Q is the initial concentration. So that's the typical propagation process. By this way, we can calculate what is the concentration level of these bacteria, and from that we can find out is there diseases or not, like detecting them, right? So this is the cut of the human body, like from the side. This is the infected area, the wound or whatever, right? Or the infected area, let's say, not the wound. I have another one case for the wound. So then you can see that they move and remors will be capturing them. So we simulate it like this. The remor is sitting here, and then this is the area that we are trying to capture, right? So now, as you see here, it takes like 2.7 hours to have this picture. So that we are showing this concentration, right, of the bacteria. So where the infection is, the concentration is very high. That's why you have this red area here, right? And then the yellow area, as you go further and further from this infected area, then of course the concentration intensity is getting less and less, right? So now what we are trying to do is also, we are trying to measure the time, how long it takes that this significant amount of quorum sensing molecules will be reaching the remors, so that remors can detect these problems, and then we can decide there is a infection or not, right? So that's the thing what we are trying to find out. And again, to do time, I show you here quickly. So these are concentration measured by mole per cubic meters. So I'm showing you varying volume fraction and varying tortuosity. And again, we change these volume fractions, alpha 0.5, 0.25, or lambda 145, 175. As you see here, that so right, really, it's a lot of things to say. But anyhow, so all these volume fractions and tortuosity are different for each of these organs, like brains and kidneys and skins, and for all the people. That's why we use these different values here, right? And then for alphas and tortuosity here. And then, so what will happen is, the higher the volume fraction, like here, 0.5, like here, and the lower is the quorum sensing concentration at the receiver. Similarly here for the tortuosity, right? And then the lower the tortuosity, the higher the concentration, the volume fraction and tortuosity, the higher is the concentration we can capture them. So that's the lessons we learned from these, studies. Oops. And then, so now another application is about, for the military personnel, how to be capture these Acinetobacter baumannii infections. Again, they're bacteria based, so they're like kind of like bacteria. They create these wounds and now they need to be somehow find a cure. And this is the #1 pathogen, require new antibiotics by the World Health Organization. So now what we are trying to do is we are trying to come up with in situ lab on a wound system, low system. That's what we try to do. And I will skip because the time is running. So what we try to do is, so this here, it looks like a box, but it's like a Band-Aid tab, right? It looks like this. So now when the wound is here, then we develop these different sensors, electrochemical reporter bacteria, same way as I showed you before, but much smaller and flat sensing units. Electrochemical without reporter bacteria, B, and the light. Again, I explained you this before. So then we will put them here, and then they can sense the information, these bacteria, and they are programmed to detect these Acinetobacter baumannii infections, or Baumannii-based infections, right? So now here are all the details you can see, and then so now again all these things. I will go to the last part because the last part is interesting to talk about for the viruses. So now we have, of course, a lot of challenges in this case too. So lab on a wound modules integration, experimental testing is an open issue. So now the last part, that's why I was running, because I have like I got 5 minutes maybe, right? Or maybe it's I need 5 minutes, guys. I'm sorry. I don't know. I spent too much time. So I tried to tell you about the viruses. So you can see there is a clear difference between bacteria and viruses, right? So typical differences are the bacteria are larger, like 1,000 nanometers. Viruses are much smaller, like here you see here, like 10 to 40 nanometers. And the bacteria are living and viruses are non-living. So they have to always go to a cell and stuck to them, and then they will suck that cell, and by this way they live. So that's a difference, non-living meaning, they always need some cell to bind. And infections are localized, and viruses are systemic. They are able to reproduce by itself, and then the virus always needs a living cell to reproduce. Otherwise they die, right? So that's why I kept telling you. And some bacteria are beneficial. We always have bacteria, right, in our guts, etc., right? They're helping us. Viruses are not beneficial, absolutely not. So bacterial infections can be cured with antibiotics. Viral diseases can only be prevented with vaccines, or they give you steroids, right? But that's not a cure, right? So most of the prevention, right? Vaccines. And examples are like Staphylococcus aureus, Vibrio cholera, and all the other ones, right? Bacteria-based. I gave also 3 of them before. And here we have HIV, influenza, and SARS, and COVID-19, and all that stuff, right? So these are typical examples. So now what we try to do is we need to restructure our work, that the concept is there, but instead of bacteria detection, we have to detect viruses. Of course, good luck, right? So that's the problem. So the most reliable virus detection in the laboratory is done by these PCR process, polymerase chain reaction, right? So, so there's this RNA, it's called ribonucleic acid replications, and then they take your saliva or some, your spit or something from your nose, and then there's one DNA, and then they can measure it, and then they keep replicating it so that they will understand more and more. And that process is long, and it needs a lot of machinery, meaning processing, etc., right? So, and then it's also not, suitable for in-body applications. So now we need to come up with alternative sensors, like new design, like byproduct sensors or antibody sensors. This is a huge area that's really research needed, because the viruses hijack the infected cells' molecular machinery to replicate themselves, right? And these sensors monitor the excessive byproducts of this replication, such as proteases. And antibody sensors, immune system recognize viruses by their antibodies binding to viruses' antigenes. And then an antibody tags a virus or an infected cell for attacking by other parts of the immune system or can neutralize its target directly. So these are the two sensors we have in our minds to design and come up with, or they're maybe off the shelf existing, but I don't know, but they are not used inside the body, right? So now, so we see that COVID-19 has all these challenges, and we need to somehow do these, luciferase sensors and antibody sensors that is already existing to my knowledge, but not for the inside the body and like inside our framework, right? So now, what are the benefits of this idea that we are trying to do for the public health? We can early detect these infections. Okay, then you can do quarantine of these people, and also then we will try to reduce the patients which are asymptomatic, and then we can continue to monitor those people. And they are connected to the internet. We can always track the infected individuals because of this Internet of Bionan Othings concept, right? We can trace their past and present contacts and also can monitor proper quarantining procedures. So there's a lot of work to do, as you can see, and we need to work on this for longer term, but that's really not an easy concept, as you know, while these people are trying to come up with the vaccines. So we are trying to come up with this big framework that will help to all these tracking infected individuals and also track these viruses and at least avoid them, right? So I thank you for your listening. I'm a little bit above the time, like 5 or 4 minutes, I can't remember now, because I use a couple of minutes to talk in Turkish, as you know. So 1 hour, I hope you got some of the ideas what I explained to you. So if you have any questions, please feel free to ask. You can ask in Turkish. I hope I can understand you. So please go ahead.
Hocam, öncelikle çok teşekkür ederiz. Hakikaten çok ufuk açıcı bir sunumdu. Çok iyi. Hakikaten hani daha neler yapılması gerektiğiyle ilgili çok güzel bir sunumdu. Çok teşekkür ederiz. Hocam ilk sorumuz şöyle geldi. Bu cyber space tarafında oluşabilecek ataklar konusunda bir çalışma var mı? Yani ne tarz ataklar gelebilir buraya veya bununla ilgili bir simülasyonunuz, anti atak çalışmaları var mı? Böyle bir sorumuz var.
Şimdi zaten o independent of this concept, independent of this application, there's a lot of work on this side, right? Like in terms of aggregation processing, also for the security purposes, independent of this particular application, right? So like all the denial of service attacks, like privacy attacks, etc. So it's already existing. So all you need to do is you take those existing solutions and try to apply in this application case. And especially the privacy is extremely important here, and also the confidentiality, right? Like the data, etc. And either you can use those existing solutions, or you can come up with your new solutions. So mostly what the world is going to is, again, I'm not into security, but the people are using more and more machine learning algorithms, combining them with the existing security algorithms, or they come up with integrated solution, machine learning based security solutions, and those can be revisited and used for this particular applications. Because do not forget, in this application, we get a lot of data, like like terabyte of data. Because if we collect information from every individual in the world, not in the world, but also in, let's say, in Istanbul, there will be a lot of information here. So then you have the scalability problem. You need the scalable, secure solutions, for example, right? So these are the things that, if people are interested in this type of research, they should revisit the problem, saying, okay, what's existing out there in general, and then what is different here in this case? I gave you some examples, let's say the scalability problem, and also maybe combine them with the machine learning algorithms and come up with some solutions, right? So there is a need for those type of solutions. I mentioned this also somewhere here, like this part 3.4, and also even hardware security. But since you're on the cyberspace, so I mentioned this to you here, securely and privately, right? Patient health data, and then also here. Then another issue here is that some of these data you cannot compress, like these X-rays, etc. So then the volume of traffic is also very high when you collect them here. So there's many open issues there too.
And another question, is it possible to remotely reprogram those bacteria you put inside the human body?
Yes.
So if so, the security risk is much more challenging than we thought.
Absolutely, yeah. So actually, that's true. Like, where is this picture here? Like, remember in the beginning I showed you this
At
picture.
the beginning, you mentioned the network is two-way. So if the network communication is two-way and we can reprogram the bacteria we are putting inside the body,
right,
we have a huge risk of security over there.
This part is the cyber world, right? So now, of course, that's really very, very important that somebody will send all the wrong information and it can create a zombie, right? All the device inside can malfunction, right? So it's very, very important that who is transmitting or who is reprogramming that, right? So of course, it's very important.
And somebody, in fact, can cut the communication with the internet and like a man in the middle attack and come to the near field communication and reprogram it. And this is a huge attack.
Yes, exactly. Yeah, of course. Yeah, that's very likely, and that's why it's extremely important, that part I kept saying about security, right, in the cyberspace. So that's an open issue, and hopefully people will behave nicely, but that's not the case, unfortunately. You're right. So now somebody can come and then really reprogram you and kill you even.
Not only you, maybe others also.
Yeah. I mean, you meaning like many people.
Yeah,
right? So
like because I am the person from the cybersecurity perspective. This is why it came to my interest.
Exactly. Yeah. Of course, yes. But also your remorse should be have high physical security, right? I mentioned this to you somewhere here. Sorry, I have to go back and forth. Physical layer security is also important, that these devices will not be here, right? Hardware security, that they will not be prone for any types of attacks also, that hardware will also be robust, that can really capture some of the cyber attacks, for example. So it will be like multiple layers of security, right?
But proceeding of, in fact, the reprogrammable devices inside the human body, in fact, may be the advantage against the viruses, because you said it should be living. The cells should be living against the virus cases. So if it is possible to reprogram those devices remotely, then this might be the main advantage against viruses, right?
So this virus case is different. You cannot program the viruses. So what you do is, be careful. You have to reprogram the cells, like let's say in this case, human cells. It's a challenge. And then how you will reprogram them, like recreate, right? Regenerate them, right? Genetically engineer them like I show you the lentil virus case. But then you will program them such a way when a virus comes and binds to it, right? And then this cell will immediately capture it and kill it, because the virus can only live through the cell. When the cell is dead, it will also die. Do you understand what I mean?
Yeah,
So,
yeah.
But here you program bacteria directly, genetically engineered bacteria. There, we are doing this cell generation and then regeneration, and then cell will attack the virus.
Okay.
And then hopefully that, that's more complicated, as you can see.
It has to find the virus cell and then kill it.
Yeah, of course. But exactly like when the virus attacks a normal cell and then it reproduces, but then somehow when it hits one of these genetically engineered cell, it can immediately start to create this colony and then kill all the around. By this way, it will stop the, reproduction of these viruses, right? That's what I'm thinking, but let's see what will happen, right? So this is when you are planning to write a proposal, maybe it's better to focus on that, like this type of concept, but more virus, right? That's that will be my suggestion, right? Virus detections and then create this framework, because we need this framework, this entire one. For example, 3 weeks ago, I received a proposal from Ireland. The company was trying to do this part, right? Like this part, meaning that from the cell phones, they will understand who are infected, because, when you're infected, you inform, right, the people. And then that cell phone information will be stored, and then from that cell phone, with whom, because it can also do location tracking, right? The system, the cell phone system, cellular system, right? And then it will find out where this person have been, and then will also inform those people that they may have been infected, right? So that was their idea, but just from the outside, right? We are talking about like internally, how can we, detect these viruses and then take precautions inside. If not, then we can also take precautions like these guys try to do, like all of these things, like, trying to isolate or detect those people who are infected, right? So it will be like multiple procedures to take care of this problem. So otherwise, the life cannot continue like this, right? Yeah. So we have to find something. If it continues like 3-4 years, the world will be miserable, right? Yeah, ya düşünün, harp zamanı 1939'dan 45'e kadar dünya felç halindeydi ya. Yani şimdi Allah göstermesin öyle bir şey olsa yandık yani. Yani 5-6 sene.
Ekonomiler dayanmaz zaten ona.
Ya, ekonomi zaten, insanlar da çoğu depresyona girer, bilmem ne, yani bir sürü şeyler. Yani inşallah bunları bir yakın zamanda bulup halletmek lazım. Ama bunlar biliniyordu ya. Yani bu virüs olayları, hatta bizim bir proposal vardı yani Norveç'te koronal demiştik. Koronal. Gene bakteri üzerineydi. Yani büyük bir center kuracaktık orada. Hatta orada konuştuk 2015'te. Ya bu korona bir vurursa yanarız diye, işte geldi maalesef. Daha bir sürü var böyle virüsler. Yani umarım dünya bu virüslerle, yani kıyamet mi derdik biz onlara? Kıyamet günü gelmez yani. Çünkü müthiş virüsler var ya. Yani bunların yani bizim aslında çalışmamız, bütün bu harp şeylerine, ne derler ona, bu silahlara, bilmem neye, bombalara vereceğimiz paraları, yani bu health veya poverty falan, bunlara yatırsak bütün dünyada Ne kadar iyi olur diye, ama işte Amerika'nın işine gelmez. Çünkü Amerika oradan müthiş para alıyor şeyden, harp şeyden, akademi diyorum, endüstriden. Ve de bakın, bütün dünyada çıkan harplerin çoğu Amerika'dan çıkıyor. Yani ben şimdi Amerika'yı kötülemiyorum da gerçek bu yani. Yani dünyanın parası gidiyor. Yani o oralara verilen paralarla dünya düzlüğe çıkardı her alanda. Ama değiştiremiyorsun sistemi maalesef. Yani
Çok teşekkür ederiz. Ağzınıza sağlık. İnşallah ama bu bir başlangıç olacak. Biz buradan çalışmaya başlayacağız. Ve sizinle temasımız devam edecek Allah'ın izniyle. Özellikle cyber
teşekkürler.
security
Kaç
tarafında.
kişi vardı ya? Ben göremiyorum. Kaç kişi vardı şeyde, TOK'ta? Bir sen mi vardın yoksa?
Yok hocam, şu anda 20 civarındaydık ama YouTube'da yayınlanmaya devam ettiği için yani gün sonuna doğru yüzleri geçiyoruz. Anlık
Oha.
olarak belki herkes takip etmiyor ama dünkü webinarda da 120'lere ulaştık gün sonunda. Bugünün
Ha,
sonunda da oralara ulaşacağımızı düşünüyorum.
anladım.
Canlı izlemiyorlar ama sonrasında izleniyor, merak etmeyin.
Yok
Ee,
canım, yani şey hiç olmazsa değsin yani.
Yok yok hocam,
Evet.
zaten biz şu anda ekibi özellikle yaydık. Onlar seyrettiler, ilgili olan arkadaşlar. Hatta
Evet.
bir çalışma grubu da başlıyor arka tarafta. WhatsApp grubumuz çalışıyor şu anda. Sizden aldıkları ilhamla. İnşallah temaslarımız devam edecek.
İnşallah.
Hocam bu süreçler bittikten sonra İstanbul'da hakikaten sizleri ağırlamayı çok arzu ederiz. E,
İnşallah.
Çok teşekkür ederiz hocam.
Ben de
Sabahın bu
teşekkür
saatinde
ederim beni
zaman
dinlediğiniz için.
ayırdınız.
İyi günler,
Çok sağ
başarılar,
olun.
sevgiler.
Sağ olun
Her
hocam.
şey iyi olsun, gönlünüzce olsun.
İnşallah hocam.
Selamlar,
Görüşmek
sevgiler.
üzere. Saygılar sunarım. Sağ olun.